In this work, we present a novel fuzzy approach towards in silico B-cell epitope identification that induces antigen-specific immune response. B-cell epitopes are located on the surface of pathogens and are recognized by human antibodies to neutralize the threat. However, experimental studies have shown that only certain regions on the antigen surface actually trigger an immune response, rather than the entire antigen. This makes identifying these specific regions, known as epitopes, critically important for many applications, including vaccine design. Identifying diagnostically useful epitopes is a difficult, time-consuming, and resource-intensive task. As a result, in silico prediction methods have gained significant attention in recent years due to their lower cost, faster results, and reduced manual effort compared to experimental techniques like NMR spectroscopy or 3D X-ray structural analysis of antibody-antigen complexes. Still, a major challenge faced by most current models is the need for large volumes of training data. In addition, many models do not achieve viable accuracy. The complex and noisy nature of epitope data makes this task even more challenging. Previous studies used structure-based methods for epitope prediction, but these approaches became very complicated when dealing with 3D structural data. Mimetope-based methods, which combine mimetope sequences from phage display experiments with 3D antigen structures, attempted to locate aligned sequences and predict potential epitope regions by mapping them back to the antigen surface. However, these techniques have not proven very effective in practice. Sequence-based techniques were later developed, but they required large, well-labeled datasets. Due to the limited and imbalanced nature of the available data, many deep learning models suffered from overfitting and poor generalization. The current work is the first to propose a fuzzy logic-based approach to in silico B-cell epitope prediction. The model's effectiveness is demonstrated using highly imbalanced and limited datasets across multiple experiments. Results show that this method significantly improves both accuracy and precision compared to existing approaches. Furthermore, the model was evaluated using the SARS-CoV-1 antigen-antibody PDB complex, where it outperformed state-of-the-art machine learning models trained on the same data. These results confirm that the proposed method delivers better prediction performance than existing alternatives. Let us walk through the proposed model architecture. First, the antigen feature vector is input into the classifier section of the model. This section performs classification using min-max hyperboxes as decision boundaries, mapping features into a high-dimensional space and calculating class memberships. During training, neurons in the hidden layer are added dynamically, which allows the architecture to update in real time. This dynamic adjustment also supports the midway introduction of new labeled data during training. The same features are also sent to a biologically inspired reflex section, which generates compensation outputs when there is hyperbox overlap or containment, reducing error in high-dimensional space. The activation functions used in the CLN, CCN, and OCN sections are also illustrated here. Next, the outputs are combined. And the final prediction is obtained by applying argmax to the class membership values to determine the class of the input sample. To train the model, amino acid sequence data and activity labels were taken from IEDB and UniProt. To reduce the impact of high-class imbalance, epitope data was simplified into a binary classification. All positive high, positive intermediate, and positive low samples were considered as positive. Despite this, the dataset still showed a significant imbalance of 75%. Therefore, we used Matthew's correlation coefficient, along with accuracy and precision, as our evaluation metrics. Here, we show feature-wise kernel density estimate plots. A weak negative correlation between some features is evident here. In the first experiment, the model was trained on two subsets, with training samples of 200 and 300, each having 75% class imbalance. Results were compared to several machine learning models. We found that our model consistently outperformed other models, delivering comparable accuracy and higher precision, even with limited data. While other models' performance varied with the number of training samples, for example, random forest performed well at 200 samples but it loses consistency and its performance declined at 300 samples, with ridge and GBC performing better. Our model showed robust and stable performance across varied settings. In the second experiment, we evaluated the model on the SARS-CoV-1 antigen sequence. Again, the proposed model outperformed all other models, showing a significant 5.77% gain in accuracy. A parametric analysis was also carried out to examine how the model's performance is affected by hyperparameters, including the hyperbox expansion coefficient, theta, and the fuzziness control parameter, gamma. It was observed that the training time is extremely short, between 1 and 5 seconds, but the sample test time is relatively longer, around 5 to 10 seconds. This contrast is not typically observed in low-dimensional classification tasks. Additionally, as theta increases, the number of hyperboxes created grows exponentially, not linearly. Training time increases sharply until theta reaches 0.2, after which it decreases exponentially for both configurations. In conclusion, we introduced a fuzzy approach for B-cell epitope identification that effectively addresses challenges related to limited data and high-class imbalance. Through extensive experimentation, we demonstrated the model's ability to learn from such datasets. Our results show that fuzzy models are especially suited for imbalanced, low-data problems. Future work could focus on reducing the sample testing time and developing algorithms to automatically tune the hyperbox expansion coefficient and fuzziness control parameters.